Welcome everyone. So in this video we're dealing with the uronic acid pathway. So what is the uronic acid pathway? So basically, just to give you a simple definition, so I can say that the uronic acid pathway is simply a metabolic pathway. So it is a metabolic pathway in which we see that there is the production of what? So there is the production of what? Glucuronic acid. So there's also the production of pentoses, and there's also the production of ascorbic acid. So ascorbic acid, you notice that it is not produced in human beings due to reasons which we are going to see as we begin with as we progress in the video. And some of the major functions of this pathway is that there is a biosynthesis of some of its functions. So we can say functions. Some functions of this pathway include synthesis of what? GAGs. So that's also among the functions, and also it also plays a huge function in what? Detoxification. Okay, so now where does it take place? So this pathway takes place in the liver. So site takes place in the what? So the site is the liver. Where exactly? In the cytosol, or in the cytoplasm, in other words. So now how does we begin? So now we begin with the pathway. So it begins with glucose. So like we said, it is a pathway which is more of a bypass pathway in which glucose can be converted to, can be oxidized, in short. So it's a pathway which is a bypass pathway for glucose oxidation. So another thing which I forgot to mention is that in this pathway no ATP is produced. So no ATP is produced. Okay, so it begins with glucose. So in the normal glycolysis, we see that glucose is converted into what? So glucose gets converted into glucose into glucose 6-phosphate. Glucose 6-phosphate by what? By glucokinase enzyme, and there's a donation of 1-phosphate by ATP. So there's a donation of 1-phosphate by ATP, so it remains ATP is converted to ADP. So it has been utilized to produce ADP like that. Okay, so now this glucose 6-phosphate is later converted to glucose 1-phosphate. So it is later converted to glucose 1-phosphate. Now you might be asking yourself a question to say, what is converting, or what enzyme is playing the role of converting the glucose 6-phosphate into glucose 1-phosphate? So it's simply a simple enzyme called phosphoglucomutase. So phosphoglucomutase enzyme, it's the one which is converting the glucose 6-phosphate into glucose 1-phosphate. So what follows next? So the glucose 6-phosphate is later converted to UDP glucose. So this is later converted into UDP glucose. By what? So there is a uridine triphosphate. The uridine triphosphate donates or is cleaved of the two phosphate molecules. So for instance, let's say we've got the uridine triphosphate, so it goes the phosphate, it has got 3 phosphates. Okay, then the glucose 1 phosphate, so this is the glucose 1 phosphate, like that. Then it loses it loses 2 phosphates. The uridine triphosphate loses 2 phosphates, so we're going to say that it will remain with 1 phosphate. So how will it be? It will be it is going to, the uridine monophosphate will get attached to the glucose phosphate, hence it will be UDP, so uridine diphosphate glucose. This is why we are seeing the uridine triphosphate here. And then which enzyme is doing this? There is a UDP glucose pyrophosphorylase enzyme, like that. It is as simple as that. So this is how far we've moved. Then what happens next? So what happens next is that the UDP glucose later it becomes converted to UDP glucuronate. So it later becomes converted to UDP glucuronate. By what circumstances? So we're having the NAD positive producing 2 NADH plus H. Okay, and the enzyme doing this is the UDP. So the UDP, so we've got the UDP glucose dehydrogenase, like that. I'm sure we are following, and understanding this pathway is going to help you a lot to deal with maybe to just be able to tell different diseases which can happen, such as the pentosuria, the essential pentosuria, which we're going to see as the video progresses. So now this UDP glucuronate has got can go to many, or can participate into the synthesis of many things, such as glucuronidase, okay, and also synthesis of GAGs, and also you can use a capital letter here, so GAGs, in other words, GAGs, okay, and also proteoglycans. This same UDP glucuronate, that's what it can be used for. Then the UDP glucuronate will be converted to D-glucuronate. So it will be converted to glucuronate. So where we'll have H2O passing, then will be there will be UDP. One UDP will be lost. So UDP will be lost. And then which enzyme is doing this? So we've got glucuronidase is the one which is cleaving one UDP out of this molecule UDP glucuronate. Then what follows next? So what follows is that the UDP glucuronate, the D-glucuronate becomes converted to L-gulonate. So here there is a NADPH plus H, which is changed to NADP positive. So whenever we see the NADPH, we have to understand that there is what we call an anabolic reaction taking place here. So the enzyme which is doing this is the glucuronidase as well. So we from this stage we can proceed to two ways. So it can either like this, it can proceed to this path or this path. So two pathways can be developed from this. One in which it happens in human beings, and the other which happens in lower animals. In lower animals, we are going to see the production of ascorbic acid. So let me just demonstrate. Stay with me. So we're going to have water will be given off, and then there is a molecule called L-gulonolactone. The L-gulonolactone at this particular point, there will be an O2 molecule to be given out, and then the L-gulonolactone, there's an enzyme here called L-gulonate oxidase. Okay, L-gulonolactone, so it's gulonolactone oxidase becomes converted to keto L-gulonolactone. So gulonolactone, which is later converted to ascorbic acid. So what is it that is important here when we look at this enzyme, the L-gulonolactone oxidase? This is a very important enzyme in that it helps in lower animals to convert the L-gulonolactone into ascorbic acid. So this is what is important. It helps to convert or to produce ascorbic acid in lower animals. In the human beings, the reason why we don't produce ascorbic acid or vitamin C, in other words, it's because we have a deficiency of L-gulonolactone oxidase, which is an enzyme needed to convert the L-gulonolactone into ascorbic acid. So now let's proceed. Also, let's view the pathway from the other direction. So where we've got the same the L-gulonate being converted to 3-keto L-gulonate. So here we're going to notice that there will be an oxidation reaction where NAD positive is being converted to NADH plus H ions. So now here we're going to see that CO2 will be produced, and then there will be L-xylulose will be converted to xylitol. Okay, xylitol, like that. So here specifically, we've got an enzyme xylitol dehydrogenase. One good thing about biochemistry is that it has got a lot of enzymes which you are supposed to remember. But one interesting fact is that these enzymes are easy to remember. Most of them, you're going to be seeing them with dehydrogenase. Most of them have got a similar ending, so it's easier to relate. Once you know where the enzyme or what the enzyme is acting on, it'll be easier for you to be able to recall the enzyme that you're dealing with. So the xylulose is being converted to xylitol by an enzyme xylitol dehydrogenase. And what happens here? So here there is also NADPH plus H ions being converted to NADP positive ions. So the xylitol will be converted to xylulose, which also has got another oxidation reaction. So NAD plus are reacting to produce NADH plus another H ions. Then it is later taken to what? There'll be xylulose 5-phosphate. Then this can go to the pentose phosphate pathway. And if you remember, when we were in our previous video, we were talking about the phosphate pentose pathway. There was this molecule, the xylulose 5-phosphate, which was being converted to fructose 6-phosphate to fructose 6-phosphate, which was going into the glycolysis pathway. And then one other thing which I want you to notice is the L-xylulose. So the L-xylulose. So this L-xylulose is really important. It's really important because it's the one which is converted to the xylulose, to the D-xylulose. The D-xylulose plays a huge role in the phosphate pentose pathway because it's going to be converted. The D-xylulose can be converted to, can be used to produce the RNA, the DNA, stuffs like that. And then suppose you have a deficiency of the xylitol dehydrogenase, meaning that you're going to develop a condition which we call essential pentosuria. And when you have this condition, meaning that here the L-xylulose will be most likely secreted in the urea or as urea. So mostly patients suffering or who have got the absence of this enzyme, if you test the urine for the presence of sugars, the Benedict's solution is going to test positive. There'll be a positive test in the Benedict's solution. And then this, you see that this enzyme is really important because if you don't have it, meaning that you're going to develop this condition. I think this has brought us to the end of this video. But just before that, let's have a recap. We said that in the beginning, the uronic acid pathway takes place in the liver, specifically the cytosol. and then no ATP is produced in this pathway. Then one other thing that you have to understand or to remember is that the UDP glucuronate is used to produce glucuronidase, and also the synthesis of glycosaminoglycans, the proteoglycans, and simply when you look at the definition, you discover that it is essential for the glucuronic acid production. Pentosis and ascorbic acid is also essential in the biosynthesis of GAGs and partly detoxification. So this is a simple illustration of the uronic acid pathway, and as it is, if you have any question, you can leave it in the comment section. Where you need clarity, where improvement has to be made, you can leave your comment in the comment section. This is where we end with this video. This is MedSimplifyHub. See you in the next video.